Greetings and welcome to our Discovery Forum 2022 here at the Center for Contemporary Sciences, updates on human relevant methods in pharmaceutical medicine and the legislative process. My name is Zahir Nali. I'm the Chief Science Officer here at CCS, and it's again my pleasure to welcome all of you. I hope this will be worthwhile your time. A couple of housekeeping elements before we get started. This is a recorded conversation. We will share the recording as soon as we can on our channels. Also, you will have the opportunity at any time. Please use the Q&A interface on get to them hopefully towards the end of the webinar. Again, welcome. With us today are some of the leading figures and companies in the human relevant methods, MedTech, life sciences, Emulate, Tissue Dynamics, Cell Inc., a Bico company, as well as Johns Hopkins Bloomberg School of Public Health. In the interest of full disclosure, I am also matriculated at the Johns Hopkins Carey Business School. We hope that after these 2 hours, we will all gain an appreciation of the cutting-edge technologies shaping drug development and transforming healthcare, that we will all be able to size up the key legislative progress in the biomedical innovation space, particularly human relevant methods. And I will give a brief definition of that in a minute, and we will hear that throughout the talks. And also, unashamedly recruiting you to join this movement that is demanding human relevancy across scientific research in the interest of public good, whether that being reducing the cost of medicine or improving the quality of life. For us to do that and to help us with this mission today, Alex Armento is here, President and CEO of MedTech, Danny Levner, co-founder and Chief Technology Officer of Emulate, Paul Locke, Associate Professor at Johns Hopkins Bloomberg School of Public Health, Yakov Namias is here, founder of Tissue Dynamics and Future Meat Technologies, rebranded recently to Believers Meat, so congratulations on that as well, on the rebranding. And of course, Itadele Namro Redwan, Chief Scientific Officer of Cell Inc. So welcome to all of our panelists. And again, if you are joining us now, we are at the Center for Contemporary Sciences Discovery Forum, updates on human relevant methods in pharmaceutical medicine and the legislative process. So welcome. The way we will proceed, most of our speakers will give 10-minute presentations or remarks, and then we will go into a moderated session led primarily by Dr. Paul Locke, and I'll be co-moderating it with him, and then we will get to the questions and answers at the end from the audience. The topics we will be discussing today are very important. We will be focusing on modeling of human health, for instance, particularly in the drug development process, efficacy, and safety. These are major challenges, and we will hear some solutions today. We will talk about health, about policy, about legislations, as well as cost and savings and investments. And probably this will summarize our predicament in the field, and I'm sure our panel will have a lot to talk about this. Therapeutic development is a costly, complex, and time-consuming process. The average length of time from target discovery to approval of a new drug is about 14 years. The failure rate during this process exceeds 95%, and the cost per successful drug can be $2 billion or more. Stupefying data here that we will get to talk about. Just a brief summary of what we mean by these human relevant methods, and we will go through them throughout the presentations, but they have different names. Sometimes people call them alternative methods, other times in vitro complex systems, in silico complex system. Briefly, they denote devices, systems, and research tools that have really picked up in the last 20 years, but over the last 10 years in particular, have gained particular sophistications, and they continue to evolve. They are offering us now an opportunity to model human diseases in ways that we could not before, definitely better than standard packages, certainly better than animal models. that are notoriously poor predictor of efficacy and safety, in particularly in the drug development process. In fact, Nature magazine a few years ago, this is a major publication in the field, major journal, declared that the sky is the limit when they talk about some of these technologies, whether it is 3D bioprinting of human organs and tissues or organs on a chip. So again, throughout the talks, you will hear terms like organs on a chip, organoids, 3D bioprinting, regenerative medicine, stem cell, nanotechnology, material sciences, computing, and artificial intelligence. All of those are coming together in an interdisciplinary way to help us think about health and the biology of health in very exciting manners, whether it's in drug development, but also in environmental health and safety testing, as well as sustainability development and certainly animal welfare. So now, allow me to, well, I couldn't actually resist not showing this one. This is a cover from the Namias lab. I think it's coming next week or so. This is a rendition of artificial intelligence with biology in a model of kidney toxicity. Now, this is the type of stuff that used to be science fiction 10 years ago, but now it is actually a reality, and we hope to hear more about this from the man himself. I am going to give you three perspectives now on what human relevant methods are, just to frame the conversation from the perspective of the scientific interest in the community, the markets, and the legislations. If we look at the number of publications as a function of time in the last 10 years or so, and look at things like 3D bioprinting or organ on a chip, as you can see, there's a growth in the numbers of scientific output, and that reflects the number of scientists and students and technicians and engineers working in these fields. Definitely growing, and that reflects a great interest, and that's wonderful to see, especially since some of them actually started with one orphan paper in the field. Let's look at this from a different perspective. Let's look at the interest and the appetite in the markets, and we can kind of gauge that, although simplistically, by looking at something they call compound annual growth rate, which is a return on investment. And if we look at the organ on a chip as example, between 2015 and 2020, 60% return on investment in the next 5 years continues with a robust 28 or 30, around 30%. Same point I will make for the bioprinting field, which is a bigger and more mature. Clearly, we have interest from the scientific community. Clearly, we have interest and appetites from the market. And these numbers, if we have legislative breakthroughs, as I will show you on the next slides, these numbers will be certainly revised upward. Now, let's look here at the legislation, and I took the liberty of chronicling some, not all of them, but some of them here for your benefit. Just give me a second, if you don't mind. I'm adjusting my screens. And this is, we don't have time to go through that, but there are legislations that are also bolstering human relevant methods. If we look at 2019, these are only the last 3 years, a major decision by the EPA, Environmental Protection Agency, a commitment to phase out mammalian research by 2035, in favor of these type of human relevant methods. In 2021, a bunch of major legislations were introduced in the US House of Representatives at the beginning, March, April, around that time. I mentioned the Humane Research and Testing Act that is calling for an establishment of, in fact, a center at the National Institutes of Health to develop and improve these type of technologies. The HEART Act, the FDA Modernization Act, which is probably the most relevant to us today, which enables drug makers to use these type of technologies instead of animal models that are very poor at predicting efficacy and safety when they compete for regulatory approvals at the FDA. It also gives the agency some flexibility. So these are major developments at the European Union at the Parliament level. We also have motion in around September 2021, motion known as B90425 that develops framework and plans of actions to accelerate the transition to innovation without the use of animals. Again, there's great momentum. The Humane Cosmetics Act of 2021 as well introduced in the House and Senate with the similar sensibilities, but focusing on the cosmetics field. And in 2022, momentum continues with the passage of the FDA Modernization Act 2.0, a similar one to the one in the House. And that opened the door for movement of this bill to executive action and probably even becoming a law, which will be a major development. So again, this is just a bird's eye view of the key ones. There are other developments, not only in the United States, but elsewhere in Canada and across the world. So clearly now we have a trifecta. We have interest from the scientific community, we have interest in markets, and we have growth in momentum and legislations. And these are two of the ones that I mentioned before on the slides. And for additional reading, some of what I discussed today is published here and there. The opinion piece in Truth Out, September 7. We have another publication coming out in Frontiers soon, Frontiers in Medical Technology, where I also chronicled the 12 reasons why these technologies are better at modeling human diseases. This is a publication that actually was focusing on analyzing a major study that I hope everyone will get to read. Danny Levner is here, so I'm not going to talk about this, but this is from Emulate, not talk about it too much. It shows and it addressed a solution for a major problem, which is liver toxicology, liver toxicity, showing that these type of devices and innovations can actually predict up to 87% sensitivity, which is unheard of, the toxicity in the liver of drugs that are being developed for human use, which is again remarkable, and also shows that by doing that, by integrating these type of technologies in the drug development process, particularly in preclinical testing, especially in the lead optimization stages, we can actually acquire productivity gains in the billions of dollars in that industry. And this is a common topic of interest to everyone here on this panel. I'll be happy to share with you this when it comes out. I don't know if this is a punishment or a reward, but I'm happy to share that. And I will stop here in the interest of time, leaving you just with the thought that developing relevant models is the quest of scientists throughout history, but it's easier said than done because there are so many components and factors here. But we are at a point now, as you will see from the work of our panelists, that this is moving closer and closer to reality, and we have incredible technologies that are modeling and mimicking human conditions with the promise of getting us safer, cheaper, more effective drugs much faster. So with that, I will stop here and invite Dr. Yakov Namias, after this just brief overview, to tell us about what's happening at Tissue Dynamics. I'll stop sharing here. I'll introduce Dr. Namias very briefly. I am going to be very brief with these intros because every one of our panelists has a resume 2 miles long. So Dr. Namias is an innovator and a pioneer. Of course, he's a bioengineer. He's the founder of the Grass Center for Bioengineering at the Hebrew University of Jerusalem. He is a founder of Tissue Dynamics and founders of Believers Meet as well. And he's a recipient of so many awards that we don't have time to count, and truly a force in the space of innovation and technology. So Dr. Namias, whenever you are ready, please take it away.
Thank you, Zahir, for the kind introduction and for the amazing overview. I just want to say personally that I've been that we have to mention Mike Schuller, one of the founding fathers of this field, and his amazing work in Cornell on the first rat on a chip more than 30 years ago. I was fortunate enough to work with him on when UREL was founded back in 2009, and since then the field has been growing by leaps and bounds. So today I'm happy to discuss again Tissue Dynamics, the sensor-illuminated platform for drug discovery that we developed at the Hebrew University. we are building the world's first autonomous drug development platform, and hopefully you'll see the platform at the end of the talk. Our main point is that we're using sensors to understand mechanism of action and to develop new drugs. We have a strategic partnerships with ATCC, the world's largest biorepository, as well as Beckman Coulter. And the company has been featured in CNN, FYI, Cathy Wood's podcast, and as well as selected as one of the world's leading organ on chip companies. When we're looking into this field, the first thing we're drawn to is $2.6 billion in cost and 12 to 14 years to develop a drug. We all know the numbers, and the numbers are due to massive amounts of clinical failures. More than 90% of the clinical studies actually fail due to an adverse event or an absent efficacy. And after that, even 10% of the drugs fail after they reach the market due to idiosyncratic events. The main reason, in our opinion, that this is happening is simply because of the unknowns. When in most of the cases, when we go to a clinical study, not only do we lack the predictive ability to say whether something works or not, a yes/no answer, but also we fail to understand exactly how things work. We don't understand completely the mechanism of action of a drug, and that leads to surprises. And the last thing you want to be surprised at is a $300 million phase 3 clinical study. So why is this happening? It's happening because we're still relying on small animal models that have different physiology, different genetics, and different metabolism from humans. And that leads to different mechanism of actions. And I'll be happy to show you at least 2 examples today. What is tissue dynamic doing about it? So like other companies here, we're using 3-dimensional human tissues, and we developed our own models for the human liver that is zonated, the human kidney that was just published in Science Translational Medicine, the human heart that is multi-chambered and vascularized, and the human brain. And then we're using those tissues that have human genetics and human metabolism in a microfluidic environment that essentially mimics human physiology. This is kind of the same as everybody else. What makes us a little different is the fact that we have been integrating sensors into those platforms for the last 7 years. And what you're seeing is the second generation of the Dynamics platform that we developed. This platform uses tissue-embedded sensors to essentially dynamically record metabolic activity and tissue function on a second-by-second, even millisecond basis. And we can do that in 384 well plate format. And these sensors allow us to understand mechanism of action, just like sensors in your car allow us to know that you have an oil leak or something is wrong with your tires. The analogy is actually pretty apt. We're looking at the energy balance in the cells, and it's affected no matter if you have if you have mechanical, electrical, or chemical failures. This platform over the last 7 years has done quite impressive things. So we discovered the idiopathic mechanism of acetaminophen back in 2015, as well as explained why troglitazone caused idiosyncratic toxicity after being approved, after going through animal studies and clinical studies, being approved by Pfizer and reaching the market and still causing liver failures in 1 out of every 30,000 patients. So our technology was showing a metabolic shift that nobody else could detect that was causing massive amount of stress to patients, and that led to idiosyncratic toxicity, and this was published in PNAS back in 2016. What I want to show you today is 2 cases where we're taking those understanding and taking them to the next step. How do we actually affect the market? So this is our kidney model. It's a proximal tubule model that is vascularized and based on the RNA-seq. It's highly validated, very similar to the human model. And one of the drugs we chose to look at is cisplatin. This is a chemotherapeutic. It's very effective for multiple types of cancer. It's just that the use is limited by its toxicity in the kidney. We usually give patients between 5 to 7 day dose, and then we have to stop it. Otherwise, they're going to go through an acute kidney failure. The mechanism in rats is vasculopathy, meaning you see the expansion of blood vessels creating pressure in the kidney, but there's really no evidence that this is happening in patients. And what we saw is that when you use this model in our Dynamics platform, we saw the metabolic fingerprint that you see on the bottom. Now, if you wake me up in the middle of the night, as you just did, because it's 3 a.m. where I am right now, you'll see that I would tell you that this looks very, very close to the fingerprint of a fatty liver disease. But this is not the fatty liver. So this is how fatty liver looks like. This is a valproate signature in the liver. And this is a fatty liver signature, but in kidneys. So what is it doing there? Turns out that cisplatin actually causes the kidney proximal tubule to accumulate glucose and turn it into fat. And that causes lipotoxicity. And it's a mechanism that wasn't seen before because rats, well, they don't use glucose like we do. And it turns out that you can block this by using an SGLT2 inhibitor, essentially blocking glucose absorption into the kidneys very, very fast, eliminates the lipotoxicity that we see from cisplatin. And the movement from that critical understanding to clinical data was extremely fast. So we looked at 247 patients in Hadassah, the medical center of the Hebrew University, and you can see that all the patients, the cancer patients that have been taking cisplatin, have elevated kidney damage markers and lower kidney function. But a fraction of the patients that are also taking empagliflozin, in addition to cisplatin, have very normal kidney function levels. And this is surprising because empagliflozin was given for people with type 2 diabetes or hyperglycemia. They're supposed to have more kidney damage, not less kidney damage. So this is why this study that was on the cover of Science Translational Medicine last year was featured in Nature Review Nephrology, because it's a mechanism that we didn't expect. Another study that is coming out is looking at the human heart. So this is a vascularized multi-chamber heart that we have in our system. And it's a very complex model that has an epicardium and pacemakers and very similar genetics to the human heart. And what we discovered is actually an oscillation that nobody saw before in the mitochondrial activity of the heart on a chip. Now, the interesting thing is that this is happening much faster than people realize. We don't expect metabolic function to change in milliseconds. We expect it to change in minutes, but it was changing in milliseconds. This was hypothesized to happen more than a decade ago, but the hypothesis said that at least in animals, it was supposed to correlate to the mechanical forces of the heart. So more demand for energy, more energy production. That doesn't happen in humans. In humans, what we see is a coupling between the electrical activity and the mitochondrial activity itself. And when you break this coupling using a chemical or genetic intervention, what you're getting is, for the first time, human arrhythmia on a chip. And this was very, very fascinating for us. So we went further and we looked at actually what chemicals being used can do that. And we looked at mitoxantrone. So mitoxantrone is, again, a cancer medication used for AML, but it's also used chronically for MS. And we know that it causes adverse events, including arrhythmia in patients. And you can indeed see the arrhythmia induced by mitoxantrone on the chip extremely fast. But because now we know the mechanism, we can increase the mitochondrial activity and actually recover this effect. And again, the nice thing about it is that we couldn't go back to rats to demonstrate this, because this mechanism does not exist in small animals. We actually had to use a porcine model to demonstrate this for the reviewers. Otherwise, nobody would believe us that this is actually happening. Let me finish by saying that all of the things that you're seeing right now are not in a single on a single device. We built a robotic platform that does all of that in an integrated fashion. So this robotic platform right now can run 19,000 human microtissues at the same time. And what we're trying to do is take it toward an autonomous drug development process where the robot makes its own decision and develop drugs for at least the early stages of discovery. And these are just a small summary of human-specific mechanisms that were developed and identified by the Dynamics platform. This includes a new mechanism for coronavirus toxicity that you just finished the phase 3 clinical study on, and is supposed to be published in the coming months. And this is where I'll stop, and thank you very much.
So fascinating. Thank you, Dr. Namias. By the way, I have to mention that Dr. Namias Kobi is coming to us at 3 in the morning from somewhere. So this is for our audience to know. So we appreciate you staying up so late. And I know you may have to catch a flight at some point soon, so you may need to leave us at some point. So we'll make an exception. Maybe if someone has a question for Yaakov on the results from our panel. I have actually, if no one has it. So, Yaakov, the lipolytic lipogenic balance is very interesting, right? In terms of driving fatty acid synthase and accumulating lipid toxicity. And you described that this is fundamentally different between animals and humans, right? And is that related in any way to the postprandial states, the feeding that we experience as humans, versus the feeding that we subject animals in experiment to, which is standard feed that is consistent all the time? Is there some root there for that kind of?
I wish it could blame it on our diet, but actually no, it's a difference in physiology. Small animals really rely on fatty acid to make more fatty acids. This is true for mice, and it's also true for very small humans. So when you're a baby, you are really dependent on very high fat diets for your proper metabolism. While large animals like us and pigs, for example, we are relying more and more on carbohydrates to make our fats. So fundamentally, this pathway is different. When you combine it to other effects like, very fast heart rate, for example, and flow in small animals, there are cases in physiology that we know in advance that rats and mice are going to be different from humans. And still, we use that model. And we can go on and on. So cancer is another interesting pathways. They're there, and obviously human aging as well.
Right. Thank you so much for that. If we don't have any more questions, so we'll resume our programming then now with Alex Armento. Alex is of course a friend of the organization. He's like all our panelists. He is the president CEO of MadTech. Alex earned his bachelor's of science in biomolecular sciences also, and he's in the leadership of MadTech that has been developing excellent and exciting products in the life sciences. MadTech is a BICO group as well, and truly Alex is one of the movers and shakers that is moving this industry forward. So I'm going to give very brief introductions for our speakers, and Alex, please take it away and welcome.
Thank you very much, Zahir. Thank you for the introduction, and it really is a pleasure to be with you today. I'm Alex Armento. I am the president and CEO at MadTech Life Sciences. I've been with the company since 2008. I've held various positions within the company, really beginning in the lab as a research associate here at MadTech. I was surrounded by extremely bright and creative scientists and some truly amazing technologies that had been developed over the previous 2 decades. And so over the next 4 years, I worked on a team that developed new tissue models, new 3-dimensional tissue models, and also innovative assays that use our tissue models to address really important human relevant preclinical safety testing needs. And with this experience, I truly felt that our technology needed to be communicated to a much larger audience. And so in 2012, we established a sales and marketing team to do this for the first time in MadTech's history. So over the next 6 years, myself and a small team, we spent quite a bit of time traveling across the United States and Europe and Asia, meeting with existing customers and new prospective customers who are scientists in the pharmaceutical, biotech, and personal care industries, and their feedback that we received from them from the market was absolutely invaluable. We were able to gain a full understanding of how different industries are using our different tissue models, and by understanding our customers' pain points and their existing testing processes, we were able to shift our internal product development and our assay development a bit to meet the future demands of the customers. And we maintain that customer-centric culture today. I became the CEO in 2018 here at MadTech, and last year we were acquired, as you mentioned, by a fantastic company, BICO, which is leading a bioconvergence charge, and by merging biology and technology with laboratory automation and robotics to optimize workflows, with the goal of really reducing the time and the cost of pharmaceutical drug development. And it's been a pleasure working with BICO. They welcomed us with open arms, and they've really provided us with some incredible opportunities that we're taking advantage of now, but we really hope to take full advantage of in the coming years. For those of you unfamiliar with MatTech, we are an in vitro life sciences company with the largest portfolio of proprietary three-dimensional human tissue and disease models in the world. We're an older company. Since our founding in 1985, we've really spent we've built an excellent reputation for our tissue engineering and our expertise in not only developing but also validating these human cell-derived tissue models that reduce and now in some cases replace certain animal tests in drug development and personal care product safety testing. Our tissue models are produced, all of them, from primary human cells using innovative tissue engineering technologies, and they really provide a microphysiological platform that model highly relevant and more predictive human biology. In addition to our products, MatTech, with the support of our industry and regulatory partners, which I am forever grateful for, we've developed over 30 in vitro assays that evaluate safety and efficacy profiles of active compounds and final formulations. And the outstanding reproducibility and high predictive capacity of our tissue models has led to really impressive international multi-laboratory validation studies. In fact, 5 of our test methods have already been formally validated and recognized by US, European, or international regulatory agencies as standalone replacements for animal-based tests. And our team is currently working very hard on validating 3 more of these assays to the standard, and we'll continue to do this for the foreseeable future. So MatTech and our products have generated more than 1,000 peer-reviewed papers and technical references and over 500 patent citations, really establishing MatTech's leading position in a very dynamic and cutting-edge industry. We currently employ 93 scientists, laboratory technicians, and client-focused staff between our facilities here where I am at Ashland, Massachusetts, and also a tissue production facility in Bratislava, Slovakia. You mentioned the market earlier. You know, I think that there are a few obvious trends that we believe are going to drive market growth in the coming years. And they're really led by a major focus on human relevant methods as alternatives to animal testing. Now, you can look at this from an ethical perspective. Consumers of cosmetics and personal care products have been pressuring their industry for years to shift away from animal testing. And they've been very successful. I mean, this has led to several legislative wins as animal testing for cosmetic products is now banned in Europe and India, Australia, and parts of North and South America. But from a scientific perspective, as you mentioned earlier, Zahir, historically high rates of drug attrition have really plagued the pharmaceutical drug development for years now. Your current estimates are very similar to our current estimates, 12 years, around $1.8 billion to develop a new medicine. And while the drug development process is highly complex and drug failures are expected, the accumulation of these drug failures in the late stages of development really lead to years of lost time and hundreds of millions of lost dollars. And a major reason for this is the current preclinical testing paradigm relies on animal models to guide decisions on which drugs move on to the clinic, and animals are not very predictive of human outcomes. So as the pharmaceutical industry focuses on human relevant alternatives to animal testing, and the FDA Modernization Act is making its way through the US Congress, new innovations are obviously well underway in the area of microfluidics and organ-on-chip technologies. And for those new to this area, microfluidic platforms, they enable three-dimensional cell cultures to develop microenvironments that are highly relevant to human physiology. And they allow for tissue models of various organ systems to be interconnected. And that's where we focus a lot of our work, our current work. We are focused on increasing assay throughput and improving customer user interfacing. And we believe that innovations in this area will ultimately be key to replacing laboratory animal. And so we do think that replacing the laboratory animal will involve putting all the organ systems together, and that's going to be essential moving forward. So just to wrap this up, we are very proud of all the scientific and technological progress that we've made over the last 35 years. But we're even more excited to continue innovating in this space. In fact, we've never been more excited about the future of preclinical testing as we are now, and we believe it has a very, very bright future ahead. So thank you.
Wonderful. Thank you, Alex. I'm sure we'll have many questions during our conversation on the panel. But if someone has a burning question now for Alex, maybe we can do it now. Otherwise, we will wait for our moderated conversation. I know I have a few. And now I'd like to.
Zahir, you muted yourself.
So I'd like to introduce you to someone who, again, doesn't need introduction, Daniel Lavner. Danny is a serial tech entrepreneur. He received his PhD from in electrical engineering, nonetheless, from Stanford University. And he was one of the co-founders of Emulate. He's the chief technology officer there now. And he's all about innovation and technology and leading the movement forward. So Danny, welcome. And the floor is yours. Thank you for being here today.
Thank you, Zahir. Let me just get my slides up. 1 second, my computer is not cooperating. All right, hopefully you can see the screen now. So thank you again for having us. It's a pleasure to be here. I'm going to go quickly through the first part of my presentation because I think my colleagues did a nice job motivating what we're here to discuss. I'm really eager to get to the latter part that Zahir hinted on in his introduction. Again, without spending much time on this, the problem statement is clear. Drug development is a very difficult proposition, with the cost of drug development at this point reaching about $2.6 billion per approved drug and not really getting any better. In fact, by some predictions, getting worse. And this is despite many of the underlying technologies actually becoming cheaper and faster, like gene sequencing. And the problem actually isn't in the approval process itself. It's actually what happens ahead of drugs getting into the clinic. It's the fact that we essentially put in the wrong candidates into the clinic because we can't really do a good job predicting whether the drug is going to be safe for people or efficacious in people ahead of actually giving it to people. 90 to 97% of drugs go on to fail in clinical trials, according to different estimates of different disease areas. And we talk often about the costs and the failure rates and so on, but I really want to highlight this community also to the human impact. This is actually just looking at drug-induced liver injury for even just these 4 drugs that have killed over 150 people. So not just money, this is actually having a very profound impact on people and people's health and people's lives. We'll get back to that at the end. Now, why is it that we do such a poor job? The main reason is because the best tools we have, or we used to have available at our disposal, are animal models. And as my colleagues mentioned, animals are simply different than humans. Animal biology is different enough from human biology that we end up getting the wrong results, especially with the smaller animals. Now, you can ask the question of why can't we do this testing in human cells? It turns out once you take a human cell out of the body and put it in a petri dish or other constructs, cells also start behaving differently than they do in the body and give you the wrong results. And this is the origin story of Emulate. This is where we looked at the petri dishes and our system and understood that the reason that these models give us wrong results is because cells are sensitive to the environment, what's known as a cellular microenvironment. In a petri dish, they're sensing the rigid plastic, stagnant fluid, isolation from other cell types, and so on, which is a profoundly different environment than what those cells are used to within the body. Now, it's with that understanding that we develop organ chip technology, which is essentially, if you really want to sum it up, it's a way of providing cells a home away from home so that they can still feel all those environmental elements that they're used to within the body, outside of the body, in the lab. And to give you a small snapshot, we'll get into this, but a snapshot of what these elements are, it's things like the right protein and chemical environment, fluid flow, interaction, multiple cell types, mechanical forces, and so on, which we configure carefully for each model. I'll give you a specific example, which is the historically first organ chip that we developed, which is the lung chip. And just like you're seeing a cross-section of the human alveolus portion of the lung on the left side, it's a thin interface of 2 different cell types, epithelial cells and capillary endothelial cells. we emulated that interface within our organ chip that you're seeing on the right, 2 cell types separated by a membrane, allowing fluidic flow on either side. Now, this first paper came up in 2010, so there's been well over a decade's worth of research on these kind of technologies with all sorts of impressive results in terms of particular diseases, particular models, particular new drugs that came out of this research, and so on. And I want to tell you that specifically within Emulate, we focus internally on these 5 main organ models, but we also provide our technology for the community within pharma and academia to use on their own. And they've been quite prolific with this, looking at things ranging from COVID to cancer to other infectious disease and so on. So very, very prolific industry. But I want to talk about something else right now, which is the fact that, look, there's been 10 years worth of proof points that say that these technologies work quite well. But really, until recently, there hasn't been this clincher that says, okay, this is actually quantitatively working well in a reliable manner in a way that should really appeal to pharma so that we can start seeing the change in the industry that we've been looking for. And we started doing that first by looking at a very specific model. We believe that validation has to be very specific to a particular model in a particular application. So the application that we started with is the prediction of drug-induced liver injury, meaning you have a new drug. Many drugs end up, unfortunately, poisoning people's livers, and you want to predict this ahead of actually administering this to people. So here's what we try to do in the study that I'm about to present to you. First off, we wanted to qualify this model, and we wanted to do this not just by our own metrics, but actually to choose a third-party industry consortium standard to qualify against, which is the IQ-MPS affiliate. More on this. Secondly, we wanted to actually quantify the predictive capacity, predictability, the sensitivity and specificity on this model in being able to capture drug-induced liver injury. Now, to continue, so we don't just leave it to people's imaginations to figure out where to apply the model. So actually draw this out to actually explain to people where the models fit in drug development workflows. And lastly, the who cares question. We wanted to illustrate to people why this actually matters, and particularly we'll focus on the economic lens for why this matters. So I'll go through this pretty quickly. So first off, I mentioned the IQ-MPS consortium. They gave us a roadmap, not just us, they gave the whole industry a roadmap by which to qualify first the liver model, then other models, and we use that as a third-party guideline. The key part of this qualification involved looking at matched pairs of drugs, where one is known to be toxic, but the other one is known to be less toxic. And we looked at 7 of these pairs. I'm just going to highlight data from one of these pairs. And you can see here in solid is rofloxacin, in dashed lines levofloxacin. You can see that across all 4 measures, 4 endpoints, you can see a very clear difference between one being toxic and one being significantly less toxic. Again, we did this across 7 of such pairs. And we were able to accomplish, I'm summarizing a lot of different parts, but we're able to accomplish the qualification measures established by this IQ-MPS consortium. But as I said, we didn't want to stop there. We also wanted to quantify the performance of this liver chip. So we expanded the drug set to 27 different small molecule drugs where the toxicity, the human toxicity profile is known. Most of these were taken from the IQ-MPS list, but we added more to make it more interesting, more diverse sets to cover different disease mechanisms, or rather injury mechanisms. And I'll skip a lot of the details. I should mention that this is already available in bioRxiv, but also is about to be published in Nature Communications Medicine coming out next week. The bottom line of this quantitative study is that we're able to show 87% sensitivity and 100% specificity for this model. And let me explain to you what that specifically means, because it's easy to throw around different sensitivity figures if you don't speak about the drug set or the about the drug set that they're applying to. So what I want to qualify here is that when we talk about sensitivity, these are drugs that have actually already passed screening in traditional models, including animal models. So when I say that we captured 87% of these toxic drugs, what we're trying to say is that those are drugs that 100% would have actually gone into clinic and would have made it into humans. And we were able to capture around 9 out of 10 drugs that would have been toxic in people that would have made it into people. So potentially reduce the number of toxic drugs, liver toxic specifically, drugs that made it into humans by almost a factor of 10. So it's a very, very significant reduction in the number of liver toxic drugs that actually would make it to humans if this were implemented broadly. Now, speaking of implementing this technology, there's actually multiple places in drug development where we can see this technology fitting, but we wanted to highlight just a specific one, which is here in lead optimization, as you're seeing in on this chart, ahead of animal dose ranging studies. The reason that liver chips are very useful in that part of the process is because if the liver chip shows you toxicity, it actually not only avoids bringing in a toxic drug into the clinic, which puts people in danger and of course have very high costs as well, but also using it in this stage of development allows you to avoid some unnecessary animal testing, which we would all like to do across all animal species, but especially as you get into the larger animal species or to non-human primates, this can be a very significant impact and very significant saving. Now, I keep hinting to the economic impact. We wanted to address the question of, great, you cut out, let's say, almost all liver toxicity. Is that a big impact? Is it something that we really need to worry about? So to address that question, we built a new kind of economic value model, which in fact we're actually making available to the community. So people should be free to apply this to their own models. And what this model does is that it tracks a hypothetical drug portfolio as it progresses through clinical testing and applies known attrition to these different drugs as they progress. Now, what's unique about this model is that we start from a breakdown of the drugs, but whether they are toxic and effective, toxic, ineffective, safe, effective, and so on, which allows us, for example, to see specifically the impact of a safety model. So safety model, for example, reduces the number of toxic drugs that are in this initial portfolio. The details, again, are all explained in this paper that's coming out. Bottom line is it allows us to create estimates of how a particular model with particular sensitivity and specificity can impact the economics of drug development. To fast forward, we've applied this model to this liver chip for drug-induced liver injury prediction, and we're seeing that even just this single model with its 87% sensitivity is able to generate the small molecule drug development industry as much as $3 billion a year if it were fully adopted in the industry. And to zoom this down into individual companies like Astra and J&J, if we apply them to the public figures that are available to us, we predict that adoption of a liver chip will generate a neighborhood of $100 to $150 million for each such company. Now, this was up here just the liver chip. We wanted to think ahead and say, well, we aren't stopping liver chip. Our colleagues aren't stopping at just liver. What would a set of 5 organ models that correspond to the 5 top organs leading to toxicity in the clinic, what it would look like if we can generate these kind of models with a similar sensitivity for that top set of 5 toxic models. And the economic impact is quite extreme. So this set of 5 would generate the industry around $24 billion, and for individual companies on the neighborhood of $1 billion. So extreme economic impact from these kinds of technologies is possible. So to summarize, we went through the effort of qualifying and validating our liver chip through third-party standards. We quantified its performance, and we're able to elucidate its economic impact, which is quite big. But I want to come a full circle and go back to the beginning, which is the human impact. At the end of the day, this is the biggest element the regulators need to concern about, and us as a concerned scientists need to think about is the human impact. And we saw that of the 27 drugs that were in our study, about 250 individuals either died or suffered or required a liver transplant. So there is also a tremendous amount to be gained, a very important focus to be put on the human impact here. And we invite our colleagues to continue doing similar work to qualify their own models in these quantitative ways and to use this economic value model to speak to the impact the technology may have. Now, to bring this back to regulation, This is why we think that the time is now for various regulatory legislative changes, because technology now has a foot to stand on in terms of solid data to motivate this legislative action. As Zahir already mentioned, the FDA Modernization Act has passed through the Senate recently in September, and it's now soon to be pending in the House. Europe is also following a similar route in terms of encouraging the development of these sort of alternative models. So, we're very happy of these with these results. We're very excited for this paper to come out. But the point I actually want to leave everybody here is a call to action. This is not just about us. This is about starting a groundswell within our industry so that these models leave the science bench and actually get incorporated into the pharmaceutical workflow and regulatory workflows. Thank you.
Thank you, Danny. Fascinating. Congratulations again on the paper. I'm sure we will have more questions, and congratulations to all our panelists on their wonderful work and new papers and this incredible output. Our last panelist now, before we go into our moderated session, is of course Itadeli Namro Redwan. Itadeli is the Chief Scientific Officer of Cell Inc., and she's a leader in this space of bioprinting. Itadeli received her PhD in chemistry from the University of Gothenburg, where she's now in Gothenburg as well. And she did a postdoctoral fellowship at the Gladstone Institute, became an assistant professor before joining Cell Inc. as a principal scientist, and now she is the chief scientific officer overseeing BioInc's and tissue engineering research and development there. So, Itadeli, the floor is yours. Take it away. Thank you again for being here.
Thank you, Zahir, for the kind introduction. It's a true pleasure to be here among the world leaders within the industry. Let's see if I can change my screen. So I want to start with giving a short introduction to some of the most common challenges we have in the world right now. And we kind of heard it earlier today in the first session, but if we look at the world, how it looks today, there are approximately 16 lives that are lost due to the lack of organ for transplantation, just waiting in the transplantation queue. And approximately 9 out of 10 drugs fail in clinical stages due to poor models for testing, and due to the lack of representative models, and due to mechanisms being completely different in small animals compared to humans. Also, the cost is large, and we heard that earlier too. And it's very, very challenging to support these drug development programs due to the huge loss, due to the also huge loss of the drugs in the pipeline. At Cell Inc., we are a bioprinting company, and our mission is to provide 3D bioprinting technologies, products, and services to enable the industry to create, understand, and master biology. And we're hoping that by doing that, we can actually enable the development of life-saving treatments in the future. So what you saw there is our current suite of bioprinters, and of course there are more to come, some that has been launched and are being built as we speak to be delivered to the industry. And what we want to do is really focus on providing 3D bioprinters that enable scientists to biofabricate 3D models by combining biomaterials and bioinks with the appropriate cell types, and then by building their models that can be of multiple cell types in a very specific architecture. And I've tried to illustrate the resolution that can be achieved. So if we look at the Quantum-X Bio, which is a collaboration between CellInk and Nanoscribe, also a bio company, we can obtain a resolution down to 1 micrometer. And moving forward, these are the light-based assortment, which relies on light for cross-linking the bioprinted structures. And on the right-hand side, we have the extrusion-based printers, which rely on air pressure. And as we move along this scale bar here, we see that we can achieve larger structures. And of course, we have a few nice surprises coming up in the future where we hopefully can even achieve larger structures than that, but also combining various technologies. So looking at the bioprinting industry, it's been a great honor to be part of the bioprinting community for the past 5 years. I've seen the application field grow tremendously, where we have customers really working in traditional tissue engineering to understand mechanisms, but also to allow the development of biofabrication methods to create structures that can be used for regenerative medicine. We also have disease modeling for drug development. Organ on a chip has also become a very common field for us. Bioprinted food with combination of biomaterials and cell cultured cells of appropriate different sources, and also cosmetic testing. So we know that none of our organs grow flat. So many of the customers or scientists that we have to talk to out there wonder, why use bioprinting when my assay works in 2D? But if we look at some of the most common tissues that we have in our body, we know, for example, that vascular cells or vascular networks, they never grow flat. They really need to grow in 3D to be able to support the tissue of interest with nutrients and so on, but also to remove waste products. We also know that stem cells and stem cell development really relies on 3D architecture. So from the stem cell differentiation and during the differentiation, as the cells organize themselves and produce their ECM molecules, which are the signaling molecules, these signaling molecules tell the cells how to differentiate further and makes up the final differentiated mature tissue. And that also has to do a lot with the 3D architecture. Looking at cancers, no tumor grows entirely flat. And to be very frank, it contains of multiple cell types, and the tumor environment is quite complex. So having bioprinting and combining various cell types with various materials can help us create more reliable models. And also for the liver cells, it is very well known that some of the most important functions and phenotypes of the liver are really relying on the 3D architecture and also the different cell types and the organization of those cell types. So what we can do with our technology is we can actually take the cell or cells of interest and mix them, of course, expand them to a certain population if it's possible to expand them. But we can also use primary cells for bioprinting. Once the cells, we have enough cells, we can actually select the appropriate ink, mix them with the cells, and introduce the cartridge into the printer and start dispensing our bioprinted construct in the desired architecture, but also in the desired cell organization and cell combination and material combination. After the printing process, there are various mechanisms for cross-linking what we biofabricate, and then the bioprinted construct can be cultured as we do traditionally by changing media and incubating, and finally analyzed using traditional methods. So I want to shift gears a little bit and show some in vivo, in-house data acquired from our lab. And in this case, the team were evaluating a bioink that we were developing for endothelial cells, and more accurately for endothelialization and vascularization of bioprinted tissues. And what we saw when we had mixed our cells with the developed bioink was that we achieved a beautiful vascular network growing in 3 dimensions and really trying to grow to reach all the cell types that were in the construct, compared to in 2D, where we see that the green cells here, we had the green fluorescent protein tagged to the endothelial cells, and we see that the endothelial cells are just lying rounded up and quite flat. Another example is, and we've talked about the importance of having alternative models for skin, we also work on developing advanced skin models. And we are, of course, not the only ones. Our goal is just to do the proof of concepts for the industry, and then, of course, have our customers and collaborators really advance the technology. But in this case, we had developed a ink based of nanocellulose and components very specific for the skin tissue. And we selected the different cell types of the skin, and we bioprinted the skin construct. So what you see here is the bioprinted constructs between, it's probably and most likely at 4 weeks of a culture. Another example I would like to show is a challenge that we received from a medical doctor at Hanover Medical School, and he was working with differentiating iPS-derived cardiomyocytes, and was really challenged by having these cells survive for a longer time, but also to beat for an extended period. And we developed a laminink-based ink as a proof of concept, where we actually showed that compared to where his cells died off after 7 days, we were able to keep the cultures alive for more than 3 weeks. But to be honest, we decided to end the culture at 3 weeks, since it was just the day before Christmas, and we felt, and also had the support from the medical doctor, that the proof of concept was actually achieved, and he was very enthusiastic about the results, and has actually now implemented bioprinting in his lab, where he's now creating more advanced cardiac models for disease modeling. And with that, I would like to thank you for your kind attention.
Thank you, Itadele. Thank you. Fascinating. A lot of work, a lot of products coming down the pipelines. So wonderful to see all that. Now, I think without delay, let's move forward to our moderated conversation chat session, where we can ask some questions and bounce some ideas together. Dr. Locke, why don't you lead us in that? Actually, let me introduce Dr. Locke properly too, because Dr. Locke is a lot of things. Dr. Locke is a scientist, he's a lawyer, he's a public health expert. Dr. Locke is an associate professor and the co-director of the public health program in environmental health sciences at the Johns Hopkins. And he's a graduate of Yale University in public health, and has a doctorate degree in law from Vanderbilt University. He's a writer, author, and thinker on public health issues and environmental health and law related to animal welfare as well as non-human relevant methods. And then we can go on, but Dr. Locke, please, take it away and welcome, and thank you for. Dr. Locke is also part of our advisory council.
Yes, thank you very much, and thank all the speakers for the really great presentations. I should just do a sound check. Can everyone hear me all right?
Yes.
Oh, okay, great. So my first question, I'm going to focus a little bit on the legislative piece of this, which Zahir started with, and I want to go back to it. And at least one of you mentioned the FDA Modernization Act, which is very close to being enacted into law, and that would remove a section of the Federal Food, Drug, and Cosmetic Act that requires animal testing and replace it with a term, I believe the term is non-clinical tests, which would expand the idea of what you could use. And you wouldn't have to use animal tests. You could still use animal tests if they were the best, but you could use, of course, non-animal tests. But some of the other laws that bills that are out there, there's a bill that would create a national institute centered around alternatives. Excuse me, there are other bills and other things that have been put into place. For example, there's a new agency that was funded at the level of $1 billion, the ARPA-H agency. Dr. Akhtar and I wrote an editorial about. So seeing that we're coming up in the season of gift giving, I'd like to ask all of you if you could get a gift of one of these legislative programs, what would you like to see? Or maybe several of them, especially thinking about how those could be used to advance the great technologies and the great work that your organizations and other organizations like you are doing. This is your chance to play lawyer. Yeah.
Let me volunteer. So I understand you're saying beyond the couple of programs that you mentioned, right? What else would we want to get?
Well, yeah, several of those programs are also they're bills, so they have not gotten, and none of them have been passed into law. So any of those sort of methodologies and techniques, what do you need from the legislative and regulatory folks to really make things run more effectively from your perspective?
Yeah, so look, it's the FDA Modernization Act is one that we paid a lot of attention to because, as you said, it removes a requirement that was probably outdated, right? Because there are many cases now that we understand that animals are less good than some of the other alternatives, and yet the existing law requires the use of animals. So we do see it as a big step forward to acknowledge that animals are not the end all and be all, or often the wrong thing to use. And we are very hopeful that goes into effect. That said, it's great that it opens the door, but we'd like to actually walk through the door. We want to make sure that it's not just we should maybe consider some of these alternatives, but actually to put some fire behind it because they matter. I mean, very often we get a question of why should I do this instead of animals? Well, there's decades of data that show that animals most often mispredict, most often give you the wrong result, which doesn't only cost a lot, but also puts humans at risk. It should be, in our view, of course, again, a great first step that we open the door to these things, but this is something that we should be actively investigating. Situations where these alternative technology could actually significantly reduce risk to people should be actively pursued.
Great.
Yeah, I agree. I think an agency within the government would also be very helpful. I think the validation process of these new alternatives are going to be crucial for gaining widestream adoption. And I think having an agency involved in driving the validation forward, we have quite a bit of experience validating models through ISO, validating assays through ISO or the OECD. And these are, as they should be, they're very rigorous validations. You're talking multiple continents, multiple laboratories per continent, a huge number of compounds being run through and developing a prediction model and then validating the system against that prediction model. Some of our OECD validations, it took 10 years from start to finish and cost well over $1 million each. So I think having some sort of agency driving this validation process forward. Obviously, we need to know the criteria that is acceptable by the accepting agencies, the FDA, for instance. Is 87% sensitivity, is that good enough, right? Is 82% good enough? and then bringing all the interested parties together, obviously the industry, the model manufacturers, and the testing facilities together to kind of streamline a process forward. Because, you know, I think if each assay is going to take 10 years and over $1 million to move forward across the finish line or through the door, this is going to take a very long time.
Any other comments or thoughts about the sort of what you need from the legislatures? And the other thing, I guess, it's that I think is raised by your comment, Alex, is clearly this is being done in other places, and it sounds like I don't want to put words in your mouth, but it sounds like from what you're saying, there is a useful model that is being used in the EU. And perhaps that's something that could serve as a basis and be exported to other jurisdictions. And to the extent that our three experts have experience in other jurisdictions, I'd be interested to know what those experiences are. Are they good? Are they bad? And you know, what can we learn as an international community when we think about validation and regulatory acceptance?
Any volunteer? Daniel, why don't you volunteer again?
I'll be voluntold. Look, we have some experience outside of the US, but I don't know that the situation is fundamentally different, in the sense that both the US and Europe have programs for qualifying our types of models, but they require the sort of the push to stay on our side, which you know possibly makes sense. But nevertheless, you know, there needs to be, in our view, some urgency, right? Again, as Alex very nicely put, we can't wait 10 years, time 10, you know, over and over again. There needs to be some fire in the system to actually get these systems approved, because people's lives are on the line.
Or some mechanism to quickly accept data that was produced outside of the validation, you know, the government committee, for instance. So we do a lot of work with our industry partners, and we get a lot accomplished very quickly going that route. And so, you know, to have a better understanding of what level of rigor we need to put into these validations to be accepted by the FDA or the EMA would be very helpful.
And Alex, if I follow up on that from your experience internationally, and maybe Danny too, and Itadelli, the differences in markets, say China versus the European Union markets versus the United States, how or other parts of Asia, how was your experience in that in terms of regulations and standardization and common data elements for your products?
Yeah, so most of our validations are through the OECD, which is you know, an organization that is accepted by multiple countries and multiple continents, and so you know, when you get into the individual countries, then it gets a little trickier, because now each individual country has the ability to either accept that OECD test guideline or not accept that OECD test guideline. We find that the US and Europe are very well aligned. But you mentioned China, to be specific, you know, that they have a different approach, right? They take they have a different approach than America and Europe, and so when it comes to testing of cosmetics, for instance, although we have OECD test guidelines that are already validated and approved, you know, China is very strict in that the testing has to occur in animals in their country. And so the data won't be accepted from outside of their country. And so it gets very complicated when you're talking about individual countries. But the OECD, I think, does a great job of at least being that umbrella organization to provide guidance to anybody who wants it.
Mm-hmm. If I can add something when it comes to 3D bioprinting and biofabricated structures, we have a huge challenge due to the fact that some of the governmental bodies are using very, very old assays to assess, for example, biocompatibility, but also some of the tests that are performed do not assess the true thing and do not mimic the bioprinting process. Also, for biomaterials, there are a lot of challenges to get those approved with the current processes. And so this is something where we've come together with a lot of customers where we are really involving the governmental bodies early on and trying to work together with them to show that, you know, we need to change the way we assess biocompatibility. And I'm really referring to constructs that are used for regenerative medicine applications, but where we are together trying to optimize current methods and at least propose alternative methods that can be used for FDA testing and approval later on. And also we've seen that there's been a lot of biomaterial and tissue engineers who have been working with bioprinting being recruited within those bodies as well, to have that expertise and knowledge within the governmental bodies, whether it's in the US, but also in Europe, because we see that the way we do treat disease has changed and will change. And hopefully 3D bioprinting and biofabrication will become more relevant in the future. So we need to change our test methods to adapt for these methods, or at least add alternative methods that are actually representative of what it is we want to measure. So that's one of the, that's just an example of how we see that there is a challenge ahead of us at CELLINK at least, and our customers.
Interesting. Itadeli, a question for you here that came through the chat. I think you answered it, but for the benefit of our listeners, from Sumita, using tech today, are we able to build bespoke tumor drugs using patients' own bioprinted cells? So it's a technical question, very important question. Speaking about individualized medicine, personalized medicine in these kind of bioinks that you develop, can you do one for, you know, any on demand, on request?
Yeah, so we do have collaborators. For example, one company based out of the UK region, which is called Carcinotech. But basically what that company does is that they do take cancer biopsies from the hospital and do create advanced cancer models specific for that patient. So they take the heterogeneous tumor, which is minimum 5 cell types as they've counted, and mix it with appropriate biomaterial and print very reproducible structures that can be used not only to identify which drug is active for that patient, but also at which dose. So that's one example. We also have, and that's for, it's become a common model. Now what we need to do is really do the validation together with the clinicians and compare it side by side by the methods used today to show that this method is stronger, or at least more representative. We also have a team in Italy who've actually entered bioprinted structures within the clinical tests. These are patients who suffer from blood cancers, and they've come very far, and their last resort is really to find the drug quickly. And this is a medical doctor, Alessandra Balduini, who's really creating personalized models for these patients to identify which drug that works quickly so that that patient can be put on the correct treatment as soon as possible. And of course, there are many, many examples, but you know, we have still a challenge to convince the clinicians to really enter the bioprinting into their clinical practices. But there's a lot of amazing work going on within that industry.
What is the pushback that you receive usually from any sectors?
I
What
think,
kind?
I mean, without going into details, but we see that the medical doctors are quite common of using traditional methods that has been used years for years and years. And there is a hurdle to make them accept new models or new ways of doing drug identification for patients. But even for disease modeling, it takes time for medical doctors to adapt new technologies. But if we look at the pharmaceutical industry, they've been quicker at picking these new technologies up because they know it's important to have more advanced models which are more representative, but also they have the courage and probably the funding to support campaigns to have better results, and of course save money in the long term by having more representative models of the human physiology. So there is a mindset, I would say, between the different expert areas.
And it's always, what Danny said, actually bringing it back to the person, right? Bringing it back to the individual behind these numbers, just not just data, but real lives and real futures and opportunities. So I think if we think about it from that perspective, and then the individualized business will become more humanized in so many ways. There is a question that's coming here, Paul. Just let me get this one in because I don't want this point to be missed. We from Alan, we also need to deal for validation. I'm reading it as it is with animal data, even if the target is human. It's pretty much complicated, the whole thing process. Yeah, of course. But what the panelists were saying is that these development in legislations like the FDA Modernization Act and others will remove that shackle in terms of doing these. Go ahead, Paul.
Sorry, I didn't interrupt you. I wasn't clear on what that question was. If the question is, should we be validating to animal data? I think that's a different question. Right. And that was something I wanted to ask our panelists. I mean, if you're confronted with that as an issue, that's a real philosophical question. And also, our person who asked the question, is that what they were intending?
Is there a last name? Yeah, that's Alon Alonso. He's one of my colleagues. I'm sure he's asking about the challenge one of the challenges that we have is when we do go to validate a new method, we do have to validate it against something, and that something is typically the gold standard or the existing animal assay that's out there currently. And so one of the challenges is trying to, well, it's not, we validate our human model and assay against an animal model, and obviously they're going to be different. And the question is, are those differences, are they true? Are they supposed to be there, or are we trying to mimic animal responses, which obviously we're not? And so, with skin hazard assessment, for example, one of our validated assays, we weren't 100% predictive according to the gold standard, which is the rabbit Dre's test. And we go back and look at some of these materials through human patch testing, and we actually, our data was correct. So there is this translational quagmire that we're in, and it's something that we're thinking through now, and I think that there's going to need to be, at least for a short period of time, several in vitro models or assays that are using animal cells, and so you can take the in vitro rabbit data, the in vivo rabbit data, translate that to in vitro rabbit data, which then could translate to human in vitro data, and then and so on back to the human.
Yeah, just to echo what Alex is saying, we're seeing the same thing, is that, we develop a human model, and then very often we come up with a question of, okay, now can you validate it in a rat version? And we don't typically have a head-to-head of human chip versus a rat animal, but they ask us sometimes to make animal versions of our chips. And then what we try to say is like, listen, that's a different model, right? It's different cells, it's different, protocols that need to go in there. The question, it's like you develop a human diagnostic and you're saying, can you use it to analyze, redevelop it to use, veterinary market? It's a redevelopment. It's not the same product. So there's definitely some set of conversations going around that where where I think people on our side of the table are saying, hold on, we focus on human, why are we going back to animals here?
And I think it makes some sense, right? So for the people that I talk to, their point of view is that if your animal in vitro method matches the animal in vivo, then we'll have more confidence that your human in vitro method should match humans in vivo. You know, I find that the best way to move this forward is to validate a human assay against retrospective analysis of actual human data. And so there's limited data available, but there's enough for some assays to get some substance out of it.
There's a follow-up question in the chat. What is the scientific value of validating an animal model if it's already proven effective in human models? I think, Alex, you've answered that partially, but I just wanted to ask the question if people would like to add on to the answer that you already gave.
Yeah, I think the animal models are really providing the whole system, connected the whole body, and it's the first resort before entering into humans where we're actually exposing humans, and then there's of course the ethical question, but animals are animals, but really it's the first time we actually have a living organism, full body, all organs with the blood system and all of that being tested.
But
so
unfortunately,
unfortunately,
forgive me to interrupt, but as Jakob was describing, there are entire mechanisms, entire pathways that do not exist sometimes in animals, but they exist in humans. So actually we'll be going down a rabbit hole. So so we have to be very careful, in my opinion, in how we approach that particular tricky question. Because, you know, of course, an animal, a mouse is a mouse, but a mouse is not a human. Give you an example with something we are all familiar with, COVID-19. These mice had to be humanized. I mean, they don't have this converting enzyme, angiotensin converting enzyme, so they cannot be infected with COVID-19. So we had to overexpress COVID-19 in the mice so that they can be infected with it. Is that not artificial? Is that testing in a real model? That's creating a hybrid model is different. So there is entire mechanism in non-human primates, same thing. These are the closest to humans, but there's not one non-human primate dying from disease burden of COVID-19. There's no multi-system failure, right? Unlike humans. So again, I mean, this is the whole point of these systems, and I think as the multi-system, multi-organ connection becomes really sophisticated and advanced, and as we can add immune core components to that whole thing, and as we develop a whole organism in these chips or similar devices, then that probably is the better way to go.
Absolutely, and I couldn't agree more. I couldn't agree more, Zahir. But this is the reality today of why it's still being used. And we also know that, of course, there's amazing science and amazing advancement going on where we have human on a ship models. Some companies are working on, but to be, it's going to take a very, very, very long time until we can actually create an entire organism, because it's not easy, and
absolutely. I cannot agree with you more. I had a dean one time at Harvard Kennedy School. He used to always say, it's only us. It's only us. I never understood what he did. It's only us. Really, it's only us. It's this group. I mean, we have to depend on. I mean, this is the leader. This is the spearhead, and we have to keep pushing forward. So if we think of it from it's only us, and we are the change makers. We are the ones who are going to push this forward with partners, with all like-minded folks. I think otherwise, we will always fall back on animal. It's there. We have tens of thousands of animal models waiting. If you go to, I mean, this is a little bit embarrassing. If you go to these places that provide animal models, and I was an avid animal modeler myself when I had my lab, and so I'm not. But it's like going to a department store. You can select any animal with any strain and different thing. That's not, that's eerie a little bit. And they are available, but we need to change that mindset. I think we need, with the advancement of these type of technologies, we need to think of, how are we, creating, solution to the gap that exists for that particular question using these human relevant methods and that type of thinking.
Perhaps.
I mean,
Oh,
movement has started. We know that the movement has started, not only in the research labs and in the industry, but the movement is here and it's real, and it is by us coming together, combining technologies, sharing knowledge. And of course, I think we will hopefully get there, hopefully sooner than later. But of course, we know it's challenging. And although a lot of us, and maybe all of us here, are convinced that we should avoid and exclude animal models, I don't think it will be that easy to convince everyone. Hopefully, we will get there.
So the change will be gradual. Is that how you think? I mean, the panelists, how do you see the change happening? The deviation, the reduction. I mean, there are structures and there are policies and there are roadmaps, and many smart people are thinking about that, and they are well established in terms of refinement, reduction, all of that. I mean, how do you see this change happening?
We spend a lot of time thinking about this. There are some others in the industry that jump right into a statement about, eliminating animal testing or whatnot. That's not, we don't think that that's realistic. Maybe as an aspirational goal in a certain number of years, we definitely want to see that. But there's coexistence in the near future, and the coexistence is important for a variety of reasons. Animals do have things to teach us, as we just discussed, but chips or other alternative models also have a lot of additional data to provide us that animals are not. So perhaps at first you have total coexistence, keep doing the same animal testing you're doing today. Maybe the second step is drop, currently FDA requires 2 animal species. Maybe the next step is you drop the more complex, more expensive animal species, but you still do it in rats, for example, and they'll ultimately eliminate animals. So definitely a graded part. But I want to focus on something that's sort of on the sideline here. We keep focusing on the regulators, and of course that's very important, because at the end of the day they're major gatekeepers here, but they're not the only gatekeepers. Pharma can already use some of this technology today without regulatory approval in order to make the better selections of what will go into their IND workup. And there are roadblocks there too. And I'll give you one that actually spans the sort of crosses between industry to regulation. So one of the questions that sometimes comes up is to say, look, we're afraid, let's focus on safety, right? Something like our liver chip or drug-induced liver injury prediction. People will sometimes tell us, hey, we like your data, but we're afraid to do it because if there's a finding that is not in line with animals, what do we do? What do we do? Then we'll have to investigate and see if we believe it, if we don't believe it, and so on. But then we'll have potentially an obligation to report it to the FDA, and that scares us because this is a system that we just don't have a lot of experience with. So there are industries, for example, where they've come up with safe haven regulations to say, well, look, we want to encourage you to experiment with this technology without the fear of repercussions that might be too costly for you. I think that's another topic we should raise for a discussion. To what extent can legislation reduce some of these fear points for industry in order for them to start, working with technology?
What are those fear points? Are they litigation? Are they, as you said, I'm going to have to jump through some more hoops to explain away, if you will? Are there other fear points that you're seeing?
That's a big one. What do we do with data that doesn't fit, right? To have a chip data rather that supports animal findings to argue that they should proceed into clinic is great. Nobody will complain about one more data point that shows something valuable, but a big portion of the value here is in catching things that the animals miss. So invariably there will be something, and our opinion is, look, we're going to make the system as we have made the system tuned as well as possible, so you can trust that if you see something with the chips you should actually raise an alarm bell and you should actually investigate. But look, this is a new technology for pharma, and sometimes they want to double check and triple check and so on, because it could mean killing a particular candidate. It could mean killing the whole program at times. So it's not a decision to take lightly. And if they do this investigation to find out that they actually don't believe it and they still want to proceed into IND, now they feel like they have an obligation to report. And if they have an obligation to report, then it's a whole, potential can of worms that they've opened with the FDA. So that's a big one to address. There are other ones that are less regulator concern. Another one that we hear, for example, is that the toxicology groups, their remit is to get drugs into the clinic. They need to test drugs and clear them into the clinic. So when you offer them a technology that actually allows them to prevent bad drugs from going to the clinic, they say, look, why? Nobody's asking me to do this. I'm actually essentially penalized for taking fewer drugs into the clinic. There's a disconnect between the company and sort of patient-based incentives versus the incentives lower down the organization.
I have a couple of comments. Dan, you mentioned the lack of trust maybe in these new methods because they're so new. I really think that's where a rigorous validation comes in, right? If you could prove that your assay is 95% sensitive, overall accuracy with a large set of compounds, then I think you can start to build confidence there. It's kind of like a two-sided coin, though, when it comes to we get the question from some of our customers. We got this data in a rat, and we got this data in a primate. Which one is right? And there's two opposing data sets. And so, again, it comes to validation and just building the model the best you can and testing it thoroughly and building trust that way.
Okay. Alex, can you talk to us about repurposing of drugs, right? I mean, recently the FDA or Daniel, so there are hundreds, maybe even thousands that have already approved safety profiles existing in the database, but their efficacy was not approved for certain applications. And I think recently the FDA earlier this year approved one molecule for neuropathy that has already good safety profile, but failed in the initial path, and now it's repurposed for another indication. So how do, using data from non-human or human relevant data, MPS data, so how do you see that's moving forward?
Yeah, I mean, I think a human model assay would have to be validated, and then you could test those drugs back on that target again in a human system. And potentially repurpose drugs that way. I think a lot about this from the other side, though. How many drugs were eliminated from the pipeline because of animal data, and how many drugs could be therapeutic to humans that could be safe in humans, but were toxic in animals? So repurposing those libraries as well, I think, is a huge endeavor, but could be crucially important.
Yeah,
right, exactly. And those are available. These compounds exist, and there are probably thousands of them.
Yeah, I mean, the well plates they are commercially available. There are even companies providing them in high throughput format, so we can use automated robots to do the tests again and find the dose response and so on and so forth. So this is already going on. It's a matter of having the right model and validating the model, and then just running the screen. This is a very common thing even in the pharmaceutical industry.
Mm-hmm. And is there a type of complex syndrome that these particular applications would be perfect for, right? I mean, there are type of diseases that are multifactorials, that are not just one organ, or sites that deemed inoperable or unreachable. How is this technology helping us there in these hard places? Maybe Daniel, I know you have experience in that.
Yeah, I would maybe give a different example, which is in biologics in general, including AAV and LNP for drug delivery and so on. These systems tend to be exquisitely human specific. We use human genes and whatnot, human specific viruses. Your pharma can't use some of the simpler models altogether, and they have to use non-human primates a vast majority of the time. We've done an analysis that shows that even if you keep the regulatory minimum required number of non-human primates, pharma is using, has to use far more than that today, because you can't just go right into the regulatory studies. So there, the bar is different. We believe in general that we do better than animal models because we're human, but they're even just doing as well as non-human primates would already be a huge saving, because nobody wants to work with non-human primates. It's expensive. It takes a very long amount of time. The ethical considerations of non-human primates. So that's potentially as you said, tip of a spear area. Any of these human-specific modalities that require these more advanced models in human primates.
Can I ask another question that goes back to some of the themes that speakers have touched on? So I thought several speakers really ended with a call to action that we really need to recruit people into this field. And I think this is a very relevant question for all of you, because you're all classically trained scientists. Are we doing enough on the academic side to really train up new scientists who understand the value of the techniques that you've been discussing? And if not, what more can we do? I'm not, I am an academic. I have one PhD student who's working on MPS, so I consider myself at least, hopefully, a small part of the solution and not the problem. But if there is something more that we should be doing in academia that you see, I would really appreciate your comments on that.
Yeah, I think it's a quick jump in on that. We do a lot of outreach, and we do a lot of training and workshops with academia in the US and in Europe, because we know that students today will be in the workforce tomorrow. And so we find that the uptake of these workshops and these training sessions are extremely high. There's a lot of interest in academia right now. And so I think whatever you guys are doing is just fine.
Yeah, I agree. I mean, we've been very, very fortunate to have numerous of professors reaching out to us, wanting to send their students to our labs internationally, actually. And it's been a true honor to have them in our labs, working side by side on their scientific questions using our technology. And so this is the way we can actually learn and develop together. And also, as Alex said, CELLINK is also doing a lot of outreach. And I think, training the future generation is going to help us, of course, change the way science is done in the future.
And Paul, just one more comment there. We see that the interest from academia right now is dramatically higher, exponentially higher than it was 10 years ago. And so, I think that as the industry continues to build itself, I think that it's a very exciting industry, and I think students are excited to learn about it now. So I think with time, it's going to continue on its exponential path.
Thank you. And it's papers like the Emulate paper showing really the sensitivity and the validity of these models, and from other groups like yours and others, will excite scientists. I mean, imagine when we were graduate students, we always go to our mentors. We want to do the new technology. Can we buy this thing? Can we get this kit? Can we buy this chip? Can we? And then you have to fight, right, for the budget. And then someone postdoc in another lab is doing something exciting. You want to try to do it yourself, and it's self-propelling, right? It's self-propelling. Once it's becoming valid, it propels itself, because that's the scientific method. It's what's behind the mountain. What am I not doing that I can do better with new technology? And it's all about that. So the future is in that direction, I think.
You know, maybe I'll add one more thing. I think that there's another inflection point happening. A few years ago, it used to be that every lab that was interested would, you know, try to make their own chips, collaborate with another academic who's making their own chips, and so on. It's really time now, and I'm not just pushing your own technology. There's several major players. It's time to start picking mature technologies in terms of the engineering, so the biologists can start focusing on the biology.
Right. Yes.
Yeah, and I think
Well said.
you know, I think that's one of the bigger roadblocks than legislation or any others, is that we're in such a fragmented industry where there's so many small players in this industry. Everyone is just trying to do a small piece of the pie themselves. And I think it's really going to take more inertia to get a single hold on more of the aspects of this industry. So building the platforms, building the engineering the tissues and validating the tests, and when it's done in 6 or 7 different places, that it just bogs it all down.
Yeah, fascinating. And the context of use, right? So first, people need to be trained how to use it properly, right? That's, you cannot just send the chips or roll them out without training people. So you need training, and that happens before. In the year 2020 with microarrays, right? When I remember, I printed my first chips with, didn't know what we were doing, but we assembled a printer we got from Stanford. I was at Cushman Harbor, and we learned, and we got the techniques, and but then companies came, standardized, Affymetrix and others, right? And they and we didn't have to print our own chips, do it at home kind of thing. Right, but that happened because of these common data elements and standardization, and people came together and defined certain guidelines for the use of these chips and what's expected and what's not expected and what's acceptable standard, right? And I think now, and correct me if I'm wrong, in this industry, we still have, different companies trying to excel in their own product and validate them properly. On their own, before creating that kind of I don't want to say not collaborative, but that environment. So and that's probably something that is needed. The standardization of these assays, common data elements, so that we will not be comparing apples to oranges. And certain prioritization scheme for what's important to get out of these type of assays. I don't know if anybody has a comment on that.
Look, I think so. One thing that I actually hear from our not even customers, but potential academic users, they say, I might meet them at a conference and say, I'm overwhelmed by how many different options there are. I don't know where to start. But the reality is, and I think that some of the, some of us are at fault because we love to put up, icon charts that show how big this industry has become. But the reality is, for example, that 90 or 95% of the companies actually only offer services. They don't offer instruments or consumables for academics to use on their own. So right away they're off the list. And when you actually look at who remains in terms of sort of a solid producers, there's a handful. You maybe 5 groups you can buy mature products that you can use in your own labs from. And I'm speaking specifically in the sort of more microfluidic organ chip stuff. I'm not specifically speaking to what my colleagues here are doing. Yeah, but there's a lot of noise in the industry, and maybe just a matter of things shaking out. Maybe just that we're in that transition point in the history of this technology where we're transitioning from what you, Zahir, just described, where people used to manually make their own, chips for gene chips.
Yeah, and I remember we used to get any data you want, right? Because you can reduce the gate, and you can. So again, what kind of programming you have, what kind of software we are using, until you reach maturity in how to use these kind of technologies, you can get a completely different set of data that is misleading. And I think this is exciting to explore a little bit more on that in the future. But that will come. I think, if I remember correctly, many institutions started core facilities for these type of things. And that changed a lot of reduced the errors, reduced, because you have authorities within an institution now with trained individuals who can teach others as a service on how to practice these type of chips. And that made a big difference in academia from a long time ago. But that's how maybe something like that, a modeling like this. I know many organizations are already starting center of excellence or core facilities at certain institutions, so that they not only provide awareness and education about the power of these technologies, but actual training on the context of use, so that this will be properly disseminated. And yeah, no, fascinating. Paul, do we have any final comments? Because we are coming to a close here. I know we kept our guests and I'm trying to scan for some questions that
Well, we did have one question, but I think I can answer it. And the question was, we talked about federal legislation. What about state legislation? And I think for many reasons, at least in the United States, state legislation is probably not going to be a very good option because you really need the federal engine to move things, since that's where the regulations really are. But I would open it to the panelists. If you have a great relationship with your state and they're doing something innovative, we would like to know about it.
Yeah, I mean, I agree. I think in my opinion, what the industry needs is a set of guidelines, right? What are the rules to play within, and then go ahead and accomplish that. And I think at the state level, you're going to get 50 different rule sets, and let's see if this FDA Modernization Act can pass, right? Hopefully they can come to some bipartisan consensus here, but I think that would be a challenge by itself, let alone the 50 individual states.
Well, if you're, I think right now we're waiting on the House. So if you want to write your congressperson and push this, it would be a good time. I think it would be. It'll probably. I think they're going to try to resolve it, in this legislative session, and there's just not much left of this legislative session. So it would be a good win.
And MatTech's done that, by the way. We've contacted all of our representatives.
Thanks.
Great. I think we still have a few more minutes, but I want to just thank everybody for being here today. This has been wonderful. I know you came from all over the place, and some also from extreme time zones. So thank you. This has been very informative to everybody who's listening. We will use this as educational material to, again, spread the word about these human relevant methods, and also the word about the great work that is being done at your respective companies. So we truly appreciate you and what you do to advance the field and make these changes possible. I also want to thank our team here, of course, Dr. Aisha Akhtar, CEO and co-founder of CCS, as well all the team who worked on making this panel possible. I'm going to forget someone, I'm sure, but Sarah Feld, Mikala, Molly, Caitlin. I'm forgetting someone, but I'll get in trouble later. But so thank you so much for doing that with us. I think if would like to hang out a little bit more, then we can, but we officially, will close this session, and hopefully we'll see you again at some point with another theme and another exciting discussion. So thank you so much. And if you have a final word, please go ahead.
Yeah, thank you very much for having me. I think these discussions are really important right now, and I think it was a good one.
Yeah, thank you. Thank you for having me as well.
Thank you all.
Thank you.
Have a wonderful rest of the day or the evening.
Bye now.
You as well. Bye-bye.